Okay. All right, so we left off on Friday, basically starting to talk about the functioning of membranes. On Friday, most of our time was spent talking about the structure and maybe small-scale function of membranes. And then today, as we finish up our discussion, we're going to really give a good treatment to these various functions of cell membranes. So most question we need to deal with is this one. Which specific proteins function to transport materials? Which specific proteins function to transport materials? Because we know that's one of the 4 protein types that you find in cell membranes, along with what other 3? Labels, receptors, and transporters are one of, and then adhesion molecules. Good. So facilitated diffusion. Facilitated diffusion is still the movement of materials down their concentration gradient, but they can't freely move through the membrane, right? Because they're polar or they're too big, or if they're charged, they really can't move through the membrane. So facilitated diffusion is carried out by channel proteins with hydrophilic cores. We've already talked about this. Those hydrophilic cores, hydrophilic means water-loving. These work and interact well with water, and if water is polar, what does that mean about these cores? They are polar. And how do you get a polar core in a protein? What part of the amino acid is polar then? The R group. Good. So water uses aquaporins. There are many other channels that work with other materials. Gated channels, these are mostly for ions. Ions tend to use gated channel. These can be open, they can be closed, or they can be inactive. Okay? Open, that makes sense, right? If they're open, the ions are flowing through that channel. Closed, that makes sense. But what about inactive? What's going on there? It means it cannot be opened. Okay? So it's closed, and it's closed in such a way that it cannot be opened, at least under the normal circumstances for that gated channel. And one of these we mentioned last time, and these may be called a ligand-gated channel, meaning there's some receptor that has to bind something to open up. Or they might be voltage-gated, okay, that there's some electrical signal that will open them up. Okay, but anyways, inactive, it cannot be opened. Also, carriers. So carriers are a little bit different than channels. They're still transporters, but they aren't channels because they specifically bind something that needs to diffuse into the cell. So it's not some channel that opens up to allow materials to move through, right? Like a tunnel in a hamster cage or a slide in a play place. That's what I think of, right? When I think of these channels, I think this slide in a play place and a child just come flying down there, right? Moving down its concentration gradient. Bunch of children waiting up at the top of the slide for a turn to go. You know what I'm saying? Does it make sense? That's a great analogy, and I'm getting nothing. I understand it's Monday. That was great. At least a smile. There we go. Thank you. I appreciate that. But yeah, these carriers, they're not like that. It would be like a slide that physically grabs a child and throws it down the slide. Right? Now you really like this analogy, don't you? You're sick. It's terrible. So the thing about these carriers is they can become saturated because since they have to physically bind whatever that is that's diffusing in, if they're bound to it, they can't bind more, right? Again with the same analogy, right? A normal slide, you can get like one kid right after another. You get kids on top of kids' shoulders all going down the slide at the same time. Happens all the time. I was going to say go sit at a play place and watch, but that'd be weird. But if the slide actually had to physically grab you and pull you down, it's one kid at a time, you know? Man, that is a great analogy. So here's an illustration of aquaporin. acting as a channel. In this case, it is open and it is allowing water to diffuse down its concentration gradient. Now, since water is so important and cells work so hard to control the movement of water, we give the diffusion of water a special name. What do we call it? Osmosis. Have you ever heard somebody say they're going to learn by osmosis? They lay their head down to sleep on their textbook? It's absurd. You can learn by diffusion that way, but water moving out of your textbook into your head is not going to help you learn the material. Now, material, of course, is not going to diffuse down its concentration gradient, but at least use the proper term. Did you ever watch a movie and you're like, man, this science is horrible in this movie, but then it's a movie of talking animals. You have no problem with that, but you're like, oh man, that their science was off on that one. But the fact that they're talking to humans is no biggie. Sorry. So here's a voltage-gated channel. And so here it needs to be opened up. When it is, it'll allow, in this case, potassium to move down its concentration gradient. In this case, it's gated, right? It has to be opened up. Something has to trigger it to open that up. This specific one is triggered by a voltage shock. But another story for another time. So here's a carrier protein, and here basically the process. So there's a binding site for whatever that material is that needs to be moved across the cell membrane. And when it binds, it changes the shape of the transporter to allow that diffusion. Good example of this is glucose. Glucose diffuses across the membrane using a carrier protein. All right. Any questions? Yeah, Chris.
Is this part of like facilitated?
This is still facilitated diffusion. Since the materials are still moving down their concentration gradient, it's not requiring any net input of energy from the cell. Okay? Because you're just allowing the materials to move the direction they want to move. Now, it may be complicated that you might have to bind it and then change the shape, but that the shape change is induced by the kinetic energy of the molecule. It's not, the cell's not inputting energy to change the shape. Okay? All right, next question. Why is osmosis so special? I know you've been wondering this. You're like, we did this in lab. We watched water either enter the potato cells if we were in a really low solute solution, or leave the potato cells in a really high solute solution. And you're like, this was awesome. Best experience of my life. But I don't understand the importance of it. Well, now you will. So water diffuses like anything else down its concentration gradient. I already mentioned this. Because water is so essential for life, we give it a special name. Here's why it's so special. When water moves, the force can be devastating, right? And you see this on a large scale, on a scale we're more familiar with, right? With flash floods, or using water like from a hose to like stop a riot. Right? Have you ever watched a video of that, of the riot police spraying people with hoses? It's powerful. Okay, the movement of water is a powerful force. When this happens inside of a cell, everything is magnified because cells are so tiny, right? Because cells are so tiny, every force is a lot more important to control. So cells exert a lot of energy to control the movement of water. Cells exert a lot of energy to control the movement of water. Some cells will take advantage of the massive amount of kinetic energy within those moving water molecules and will use it to do something. Have any of you ever been stung by a jellyfish? It's great, right? No, it's horrible. And so the cells, they're called cnidocytes. They use the influx of water and the force created by that to shoot a barb out. Okay? And then that barb is what, sticks into your skin and either delivers a toxin or just, acts like a whip with pieces of glass in it that when you pull it away, it rips pieces of flesh away. Now, it's super tiny, so you don't see that, but either way, you get that irritation. And so that's an example of a cell actually harnessing the power of water rushing into the cell to do something, to harpoon something. And they don't really want to sting you because they can't eat you, but you just happen to, get in the way. You just happen to be in the right place at the wrong time. right? It's not the wrong place because you get stung by jellies in the ocean. That's always the right place to be, but at the wrong time. Wrong time. Man, that just came to me. You're welcome for that one too. Now, water will move until two pressures are balanced. You have what's called the osmotic pressure, and that's potential energy. Osmotic pressure is potential energy. This is created by water's desire to move to where there's less water. Okay, that's osmotic pressure. The pressure created by water actually moving is called hydrostatic pressure. Okay, and so water will move until the pressure created by that water moving and smacking against the walls of that cell is balanced by water's desire to move. Okay, and so if you would have allowed your potato tubers to sit in those solutions long enough, you would have stopped seeing the flow of water. Some of you even saw that towards the end, right? Where your last two measurements were identical, maybe even your last three. You're starting to see that in some of those solutions, hydrostatic pressure created by water moving is balancing water's desire to move. Balancing water's desire to move. Okay? So to sum this up, why is osmosis so special? And it's because it's a potentially devastating force to the cell. On top of that, cells need water, right? Because all of the chemical reactions that take place within a cell are taking place within a water-based solvent. Make sense? So cells need water, and the movement of water can be devastating. Which makes sense, because even on a large scale, we need water, but way too much water can be devastating. All right. So here's a demonstration of osmosis, although you all saw this in the lab. So here we've got a really high solute concentration. We have a membrane that won't allow that solute to cross the membrane. Okay? So the only thing that can happen is water to diffuse in to dilute this solution. But if this is pure water and this is not pure water, there's never going to be a point at which there's the same amount of water on either side. Right? There's always going to be more solute here than there is here. And that solute takes the place of water, right? There's something in the water, it takes the place of that water. That's why when you get into the bathtub, the water level rises, right? Like, I haven't been in a bathtub in 20 years. I don't even know what happens. Well, when you get into a hot tub with a whole bunch of other people, the water level rises, right? And topples over and, sorry. Actually, I'm not sorry. You're welcome, okay? And so this is set up that eventually it's going to get to a point where the pressure in this tube balances water's osmotic pressure or desire to move in. Gets to a point where there's enough material in here pushing against the wall of this membrane that it balances out the pressure created by water's desire to move in. Okay? So even though this will always have more solute than this, water will not always run into here. Okay? I hope that makes sense. If it doesn't, I'm sorry. Maybe it will one day. All right. We'll do one more question, then we'll take a lecture break. How can we predict when and where osmosis will occur? So this we've already seen in lab. You should be able to answer this question without going through the slide, but we're going to go through the slide. And water, like anything else, diffuses, so it moves to where it's less concentrated. And so we have this question, when, bless you, when is water less concentrated? Well, the simple answer to that is water is less concentrated whenever there's more stuff in it. And again, because whatever that stuff is, it's taking place that water could occupy. Right? If it's salt in there, it's taking place that water could occupy. If it's glucose, it's taking place water could occupy. And this is especially true because whatever dissolves in water is going to interact with the water, right? Because if it dissolves in water, what does that mean about its chemistry? It's either polar or it's ionic. Either way, it's going to interact with the water molecules. Less solutes means more free water. Less solutes means more free water. Okay, so we have a value called tonicity that provides us some information about which direction water is going to move. Okay, so tonicity is basically, it's a measurement of the total number of what we call non-penetrating solutes. Solutes that cannot cross the membrane. Okay? And so a hypertonic solution has more solutes than what is inside of the cell. And we saw in lab last week that your data should have come out to where you had 4 hypertonic solutions. And not 4, 3. Which 3 should have been hypertonic? And again, the reason you may not have got these data is because your tubers may have been too big. So it had too little surface area and it distorts everything. So 3 of our solutions should have been hypertonic. That is, they have more solute that can't cross the membrane than what's inside the cell. 0.4, 0.5, and 0.6 molar. All should have been hypertonic. Hypotonic solutions have less non-penetrating solutes than what's inside the cell. Just like you had 3 hypertonic solutions, you should have had 3 hypotonic solutions. Which ones? 0.1, 0.2, and 0.3. Sorry, you wanted to go backwards. Okay? Because the total solute concentration within potato cells is about 0.35 molar. Okay, I think most of you got somewhere around there, or maybe somewhere around 0.4 molar, and you did a good job. And you're like, well, why wasn't it closer to 0.35? Maybe our potato was a little dry, right? Those of you that went on a Monday lab, we went and got our potato from the calf. And I took what they gave me, and they gave me an ugly potato, right? I didn't get to go and pick a good-looking potato that looked nice and moist. You go over there and you ask them for a potato, they give you one they don't want to use anyways, right? And then isotonic solutions have the same solute concentration as what's in the cell. Now, in our bodies, cells work really, really hard to maintain isotonic conditions because they do not want water flowing in and out of the cell at unusually high rates. Okay? Again, because that force can be devastating. That force can be devastating. So what happens when you drink distilled water and the solute concentration of your blood drops to a point where it's way lower than your red blood cells? Water starts running into your red blood cells and starts blowing them up. That's why you don't drink distilled water, at least not in large amounts. Sometimes if you have a solute imbalance, they'll have you drink distilled water. But anyways, and it's the opposite is the reason why you don't drink marine water. You don't drink salt water because it'll raise the solute concentration of your blood so high, water will leave your red blood cells and they'll shrivel up and then clump and form blood clots. Okay? It's best to drink tap water. Or if you're stranded somewhere and your only access is salt water or something else, you choose that something else regardless of what it is. You kill an animal and you drink its blood. You know? Got to stay alive long enough for the rescue to show up. You know? Plus, I mean, it's there. You know, you don't want to waste it. It's so delicious. All right. So here's the flow of water in response to non-penetrating solutes. And so again, water is going to flow to where there's more solute. And so here's an illustration of that. Here's water put into a hypotonic solution, or a cell put into a hypotonic solution, distilled water. Water's going to rush in, bag's going to swell up. Here's it put into a hypertonic solution. Water's going to rush out, bag's going to shrivel up. And here's isotonic solution. No net change. Now notice water's still moving in and moving out, but there's no net change of water. Here's the same thing illustrated with red blood cells though. Here they are all swelled up, ready to pop. And here they are all shriveled up. And this is not good. Shriveled up red blood cells, they clump together and form blood clots. Oh wow, I just got all kinds of pictures. It's just, here's it in a plant cell. And so here's a plant cell where water is rushing in. The cell wall keeps the plant cell from blowing up. You're like, well then how can I overwater my plant cell? So there's a different thing going on there we'll talk about in a couple of weeks. Here's water rushing out of the plant cell and pulling away from the cell wall. And then here's a plant cell in an isotonic solution. Same thing illustrated in 3 consecutive diagrams, one with a synthetic cell, one with a red blood cell, and then one with a plant cell. Okay, here's what we're going to do for our lecture break. This is what I'd like for you to do. I would like for you to come up with an answer to these 2 questions. Question 1, why would a cell ever need to move something against its concentration gradient? Okay, why would a cell ever need to move something against its concentration gradient? And 2, how will the cell accomplish doing that? Okay, why would the cell ever want to move something against its concentration gradient? So that is the opposite of the way it would diffuse. And then 2, how can the cell accomplish that? All right, take 90 seconds. Does that work? 90 seconds starting now. I went to school with a guy.
So like say something like
So I tried to accomplish that with my kids. Welcome.
Take away from the outside. Or better than trying to get rid of materials.
Got about 30 seconds.
you got it.
10 seconds. Wrap those conversations up. Do you find that after I pray that God would relieve us from being, dwelling on some of those distractions, do you feel guilty then talking about some of those distractions during our lecture break time? Or you're like, no, I'm so numb. I don't even listen to you, Doctor. See, now I told you I was mean to genetics, so I needed to be mean to you all collectively as well, so that if I'm going to have bad evaluations, right, it should be across the board, right, every class. Because they're like, why does everybody hate you in genetics, but they love you in cell? I was just too nice to them. Just too nice to them. All right, so what is it? Why would a cell desire to move materials against their concentration gradient? Levi.
Cells tend to maintain a negative environment inside.
They do. Absolutely they do.
And so to do that, they would bring in ions that help.
Yeah, they either bring in excess negative ions or they pump out a bunch of positive ions. And it's usually accomplished that way. You pump out a bunch of positive ions, you end up with a negative charge inside of the cell. And that's so cool. And you're like, why would a cell want to do that? And the answer is, of course, to create an electric field in your body to make you a magnet for lightning bolts. That's not the reason at all, but it is a consequence of that. But no, that's to facilitate electrical communication, right? If the cell's normally negatively charged, then you have a really cool thing that wants to happen, and positive charges want to flow in and change what we call the polarity of the cell. Good. Absolutely. It's a great reason why cells might want to do that. Can we think of any other reasons? No, he did have his hand up with the other one. Then we'll go over.
You were saying that if the cell needs water, it'll have to pump something out. So it can get
Yeah. If it wants, well, it actually needs to pump something in if it wants water, because if you pump something in, solute concentration inside's going to go up, water's going to follow it. But yeah, water follows wherever there's more solutes. And so if you need more solutes outside of the cell, sometimes you have to pump solutes outside of the cell to get water to leave. Absolutely. It's a good way for cells to control the net water flow. Mia?
To like remove waste.
To remove waste? Yeah, absolutely. Sometimes you have a lot of materials outside of the cell, but you need to keep getting that material out, okay? Because its presence can be devastating for the internal conditions of the cell. Another one is, remember, when I told you when you see concentration gradient, you need to think potential energy, okay? So sometimes it's just to get that boulder to the top of the cliff, right? Our textbook example of potential energy, right? To get the boulder to the top of the cliff, this is the chemical version of that sometimes. Set up a concentration gradient, right? Get that boulder to the top of the cliff, set up a bunch of potential energy, because then when you open up those channels and that kinetic energy flows, you can fuel other processes with those. Like the formation of ATP, but we'll get there in a couple of weeks. All right, now how can a cell do it? Yeah.
So it
Davies.
uses ATP as the energy needed for active transport.
Yes, active transport, and it's fueled by ATP. Good. Awesome. Well, let's talk about that. So here are our next 4 questions. These are our last 4 questions. What if something needs to be moved against the gradient? How does primary active transport differ from secondary active transport? How does the cell eject large molecules, which you answered for your quiz on Friday, right? Exocytosis. And then endocytosis, you're like, wait, the quiz on Friday? There was a quiz on Friday? Of course there was, right? There's a quiz every time we meet for class. That way you don't have to ever wonder, is there a quiz? Is there not a quiz, right? Because there will always be one. Unless, of course, we take a test and then there won't be. So, I mean, unless you want one. I could put a quiz and a test. And that could be kind of fun. All right. So what if something needs to be moved against its concentration gradient? Simply put, active transport is going to carry that out. And so this requires the cell to input energy to move materials against the concentration gradient. Maybe not every single ion, but at least every collection. So for instance, 3 sodium ions can be pumped out with a single ATP molecule. 3 sodium ions pumped out with a single ATP molecule. Okay? And you're like, well, cool, like we're pumping out sodium ions, and what's the charge on a sodium ion? It's positive, and we know that because we'll look over here, and we talked about this a couple of weeks ago, and we're like, okay, sodium's right here. Elements on this side of the periodic table, they get rid of electrons to get what every atom wants. What does every atom want? A full shell for its highest energy level, what we call a full valence shell. Okay? Bless you. All right? So sodium's got positive charge. If we're pumping out positive charge, the internal condition within the cell becomes what? Negatively charged. And again, the purpose is to make you a magnet for lightning. Allows cells, so this allows cells to take in essential nutrients that it already has a lot of. If the cell needs to stockpile materials, allows the cell to remove waste products or secretory products. Maybe it's a gland cell and it dumps some secretory product outside. Maintain concentrations of key ions: sodium, hydrogen, potassium, calcium. I don't know why I said sodium first, because hydrogen's written first and is before sodium alphabetically. I don't know what happened there. I saw sodium, I had already mentioned it, I must have gotten excited. Set up an electric potential. This is what you talked about, Levi, setting up that negative charge within the cell. This you find especially true in neurons and muscle cells that use this to communicate electrically. So you can send a signal down your nerve from one neuron to the next. And then when it gets to a muscle cell, you can trigger the motor proteins, myosin, to shorten those fibers, to shorten the muscle. It's good stuff. Any questions about this? I have a picture of an action potential. It doesn't come out of your textbook, nor the one that I pull a lot of images from, which is a textbook we used to use, but it was really expensive. And the textbook we use now is free. And free is less than really expensive. So, are the figures always wonderful? No, but it's free, right? And free is less than really expensive. I like that. Okay, so here's the action potential. So here's the cell's normal resting state, negative 70 millivolts. And why is it negative? Because the inside of a cell tends to have a negative charge. Okay, something happens, some stimulus comes and opens up some ion channels, and a whole bunch of positive charges rush into the cell. Okay, we call that depolarization, and so it shifts over from being negatively charged to being positively charged, because all this positive charge, mostly in the form of sodium, comes flying into the cell. What it does is it creates a chain reaction, and so this starts happening all along the membrane of the cell. And then eventually it gets to the end of the cell and signals that cell to tell the next cell to open up its channels. And it's like, boom, this carries all the way down a neuron. So that you can think, man, I want to move my finger, and all of a sudden you're moving your finger, right? Or better yet, this one, right? Like, I need to get a ride, and I need to impress them with my hitchhiker son. Look at that, 90 degrees. It's impressive. I've never hitchhiked. That would be pretty scary. I'd rather just jump on a train and ride it till it ended, right? Just be like, yes, this is where I'm gonna live now. No? Never? You don't ever have those thoughts? Oh. Yep. Wait till you're trying to have a dinner with 3 kids under the age of 7. You're like, where's the train tracks? All right. So then you'll have a repolarization period where the cell has to restabilize to its resting point. Okay? It's recorded too. I mean, it's just, you're just like, you know what Dr. Engel said today in class? All right. So then we've got repolarization, and notice it actually bypasses its normal resting state a little bit. And this is just, like, if you're repolarizing, it's re-kicking out positive charge. And sometimes it goes a little bit too far, and we have to get back to the resting stage. Now, an action potential can usually happen every 8 milliseconds or so. You can get another action potential in one of those neurons. It takes about 5 milliseconds to have the full action potential, and by about 8 milliseconds, you can do it again. So, by the way, there are 1,000 milliseconds in a second. So if it takes 8 of these, you can have, let's say it was 10, you could have 100 of these every second. But since it's 8, it's actually more than 100. Yeah?
This looks really similar to like the QRS.
Oh, in the heart? It's just the curve. Okay. Yeah, there is the relationship because the muscle cells in your heart are stimulated by electricity. But, it just happens to have a similar shape to the curve. Yeah. Okay. All right. So how does primary active transport differ from secondary? So we just talked about active transport, and now we're like, well, how does primary differ from secondary? And you're like, I didn't even know there was. I didn't even know there was primary and second. I guess it would be were. I didn't even know there were because there were 2 of them, subject-verb agreement. I didn't even know there were primary and secondary active transports. Well, here's what's going on. So in primary active transport, the same protein that's pumping the material against its concentration gradient is also hydrolyzing ATP. It's removing that phosphate group from ATP, releasing that energy, using that energy directly to pump something against its concentration gradient. You see this used for pumping ions against their concentration gradient, which can be used to generate a membrane potential. And if you're pumping hydrogen ions, you can use this to regulate pH, right? Because pH is a measurement, it's a negative logarithm of the hydrogen ion concentration. Y'all remember that, right? We talked about that a couple weeks ago, I think, and then you've seen it before. You also see this work to store calcium ions. So in order for muscles to contract, you need calcium ions. Because myosin, that motor protein that walks along actin, needs calcium, okay? And so in muscle cells, you find a lot of calcium ions stored up inside the endoplasmic reticulum. And so to store them up in there, you have to force them to go opposite the direction that they want to go. They want to go out, but you're just packing more and more and more of them in there. So at the point that you need them, you release them, and there's a ton of them to facilitate that muscle contraction. Sodium-potassium pump. It's the most well-known pump. Pumps out 3 sodium ions for every 2 potassium it brings in. This is used to generate that resting potential, negative 70 millivolts. Okay, so that's primary. Same protein that's pumping the ions is also hydrolyzing ATP. So in secondary, it must be 2 separate proteins involved, right? So in secondary, ATP is hydrolyzed by 1 protein, and that's used to set up a concentration gradient that will then pull materials against their concentration gradient. Okay, so you have 1 protein, hydrolyzes ATP, uses that to set up a concentration gradient. And then that concentration gradient, the movement of material down, that kinetic energy can be used to move something else against its concentration gradient. Yeah, David.
What's hydrolyzed again?
Hydrolyze means to cut with water. Right, we've seen that word with our biomolecules. And so if you hydrolyze ATP, you remove one of the phosphate groups by cutting it with water. Yeah, you bet. And then there are 2 types of secondary. There's symport, where the concentration gradient that you're building up, when it diffuses down, it moves in the same direction as whatever you want to move across the membrane. Okay, whereas an antiport, they move in the opposite direction. So an antiport, you've got 1 protein building up this concentration gradient. I should have a picture of this. I was going to draw it, but I should have a picture. But you have 1 protein setting up a concentration gradient. You allow that material to diffuse down its concentration gradient, and as it moves this way across the membrane, it fuels the pumping of something this way against its concentration gradient. And you're like, wow, that seems so weird. Why would you separate those tasks between 2 proteins instead of just always using primary? That's a wonderful question. Any of you think of an answer to that? Why wouldn't you just always use primary, right? Have the same protein that's hydrolyzing ATP, just pump whatever that material is against its concentration gradient. What do you know about proteins and as they bind materials? Does it tend to be very specific or non-specific? It tends to be very specific. So this allows you to basically have the same machine that's hydrolyzing ATP fuel the active transport of maybe dozens of different materials that are all fueled and facilitated by 1 protein that hydrolyzes ATP and sets up a concentration gradient. Yeah.
Is this something you see in like a tissue, like a bunch of cells working together to do this, or is
Yeah, I mean, yes, absolutely. Yeah.
could a cell do this by itself though? Like
Yes.
concentration gradient?
Yeah, so a good example of that would be glands. So let's say you've got, we use any gland as an example. Let's say we're going to use the pancreas, right? So the pancreas does a lot. It releases insulin that helps to control blood sugar levels. It also releases several different digestive enzymes, okay? So there are individual cells that are regulating the concentration of materials and pumping their secretions out. But then you have hundreds, if not thousands of cells that are all working in unison to do the same thing. And so the cells are working both individually and corporately. Same thing happening in, say, your heart tissue, where individual cardiac muscle cells are contracting, but they're working in unison to contract an entire chamber of the heart. Micah.
So secondary active transport uses less energy?
No, not necessarily, but it's less specific. Now the protein, because you're facilitating the active transport not by a protein grabbing something, but by creating a concentration gradient that the kinetic energy is going to fuel the movement of this. So a good example of this is, it's not a great example because this is ATP is hydrolyzed, but what happens during photosynthesis. So what happens during photosynthesis, and we'll see this in a couple of weeks, actually more like 5 weeks, is there are complexes in the cell that harness sunlight and use that energy to set up a concentration gradient of hydrogen ions. okay? And then that concentration gradient will fuel the movement of hydrogen ions through one complex that allows hydrogen ions to move through. And that complex is called ATP synthase. And so the kinetic energy actually turns the machine and assembles ATP. Okay? And so in this case, it's like sunlight is fueling a process that then can actually fuel other processes. And I know it's not exactly like secondary active transport, because in secondary active transport, the energy is coming from ATP and not from sunlight. So here's primary active transport. And specifically here, I'll show you this one. Here's this sodium-potassium pump. And so this same protein is both hydrolyzing ATP. You see that there? It's cutting a phosphate group off of ATP to generate ADP. It's the same protein that's releasing the energy bound in ATP is also pumping the ions. 3 sodium ions out for every 2 potassium that come in. So 3 positive charges leave, 2 positive charges come in. So every time this pump works, you have a net loss of 1 positive charge.
And this is an example of secondary.
No, this is primary. Yeah,
Okay.
this is primary because the same protein is both pumping and hydrolyzing ATP.
Okay.
Here is secondary. Secondary, we didn't even show the hydrolysis of ATP because the hydrolysis of ATP just generated a concentration gradient. The concentration gradient is going to fuel the active transport of materials. So in this case, we are actively transporting this blue material, whatever this blue solute is. Okay? You see there's a lot of it here, very little of it out here. But the cell needs a lot of it. So it wants to keep that concentration of blue ions high. Well, what it does is it hydrolyzes ATP to set up a concentration gradient of the red ion. And then as it allows that red ion to diffuse, the red ion fuels the pumping of blue ions against the concentration gradient. And this would be symport because the concentration gradient, the diffusing is happening in the same direction as the active transport. And then here is antiport. Again, we are setting up a concentration of red ions. As we open this channel, red ions are allowed to diffuse. That kinetic energy is used to fuel the active transport of blue ions, but in this case, the blue ions are moving the opposite direction of the diffusion of the red ions. Okay? But you can see this is secondary. At no point in here does it show the hydrolysis of ATP, right? Because it was a completely separate protein that was involved in that hydrolysis of ATP. And it was setting up this concentration gradient. And the beauty is this concentration gradient may fuel a dozen processes, may fuel the active transport of a dozen different solutes that are all fueled by the same protein that set up the concentration gradient as it hydrolyzed ATP. Okay. Make sense? Questions? Concerns? So why would a cell want to move materials against the concentration gradient? To set up a concentration gradient, right? To get rid of waste products, to concentrate some material it needs, to set up the resting potential. And how does it do it? Does it through active transport, either primary or secondary. If it's primary, the same protein pumping is also doing what? Hydrolyzing ATP. If it's secondary, something's hydrolyzing ATP, setting up a concentration gradient, and then that concentration gradient is fueling the active transport. All right? So, exocytosis, endocytosis. How does the cell eject large molecules? So exocytosis and endocytosis also require energy, and most of you picked this up as you were answering this on the quiz. You talked about in exocytosis, vesicles are going to move from the Golgi body to the plasma membrane, right? And there's something inside those vesicles that when that vesicle fuses with the plasma membrane, gets released to the external environment. But we talked about this before. How does a vesicle move? What actually carries the vesicle? So, well, something walking along the cytoskeleton. Motor proteins, and specifically kinesin. And we talked about this. Every step kinesin takes requires what? 1 ATP. And sometimes it has to take millions of steps to get it to the plasma membrane, requiring millions of molecules of ATP. You're like, wow, that's a lot. That's why every cell in your body makes and breaks 100 pounds of ATP a day. Okay? There's a big demand for ATP. So secretory vesicles leave the Golgi complex and go and contact the plasma membrane. When they do, the vesicle fuses with the membrane, ejecting its contents outward. Whatever's inside that vesicle, when that vesicle fuses with the membrane, are now going to be released outside. Whatever's inside the wall of that vesicle is now going to be in the plasma membrane, right? So this is also a way in which you get protein pumps or protein receptors or protein labels or protein adhesion molecules to the plasma membrane. You put them on a vesicle, walk that vesicle out to the plasma membrane, and now that protein's there. Right? So here's an image of exocytosis. Okay, so here's this secretory vesicle packed full of whatever material the cell wants out. Might be waste, might be some secretory product like insulin or some kind of digestive enzyme secreted by pancreatic cells. Okay, and walks this vesicle over to the plasma membrane. When it gets there, it fuses with the plasma membrane, releasing whatever that product is. Yeah, Micah.
What are the proteins on the outside of the plasma membrane?
These? I mean, they're not illustrated what these are, but I mean, they could be anything. They could be transport proteins, labels, receptors, adhesion proteins.
You're showing that there's a specific spot for the vesicle to go to?
There is, but usually not because of a protein label, but because of which specific microtubule it's walking on. But yeah, I mean, it could be. But remember, a lot of the proteins in the lipid bilayer are also mobile. You know, unless they're anchored in place by the cytoskeleton. All right, endocytosis. Endocytosis is virtually the exact opposite of exocytosis, which is wonderful because exo means outside, endo means inside, right? And those are kind of opposites. You're like, why are they kind of opposites? Well, because, anyways, sorry. Not perfectly opposites. All right, so cells can trap materials in pits. And so they'll bulge a portion of their membrane outward and wrap around materials into a vesicle. So you have a specific type of endocytosis called pinocytosis or cell drinking. And so what the cell does is wraps up a whole bunch of extracellular fluid and whatever's inside of it and pinches that off into a vesicle inside the cell. You're like, that's so cool. Why is this cell drinking? Won't water just diffuse across the membrane? Yeah, so it's not to get that water, it's to get what's in the water, right? Make sense? You're like, well, then why do we call it cell drinking? Because it's not eating, right? It's like when you're drinking to get the minerals in whatever you're drinking, right? You're still drinking, but you're not really drinking to get the water, right? You may be drinking to get the minerals. Then you have another type that's called receptor-mediated endocytosis. And so this is triggered by very specific molecules. Something binds to a receptor on the cell surface, and the cell creates what's called a clathrin pit around it, a vesicle around that material. Okay? But that material is actually physically bound to the membrane. It's not just suspended in the extracellular fluid. It's physically bound to that vesicle wall. Yeah.
What happens when, as it pinches off and it pulls it into the cell, what happens to that, the membrane that's still after it opens it up and gets whatever it needs out of it? What happens to the membrane?
Yeah, it can be recycled. Recycled and then put onto a vesicle to go back out to the plasma membrane. Yeah, so these processes have to be happening simultaneously. At the same time that you have endocytosis happening, you have to have exocytosis happening as well to restore plasma membrane that you're losing through endocytosis. Yeah. So oftentimes these vesicles are going to fuse with a lysosome that then is going to either digest that material or that product may leave the vesicle. Those protein channels, those receptors that that bound whatever it was that triggered this endocytic event can be recycled and sent back out to the plasma membrane. And then this is where it's kind of cool. There are some cells that this is their primary means of getting energy, is through phagocytosis, which is a type of endocytosis. It's their primary means of feeding. So you find single-celled organisms that feed that way, and then our white blood cells feed that way, right? That's how they get their nutrients, is typically by phagocytizing foreign material and then recycling whatever's in there, digesting whatever's in there to release energy. All right, there are a few images that go along with these concepts in the last, what would it be, 1, 2, 3, 4 slides of this chapter, but that's it. So on Wednesday, we'll start in on DNA and our discussion there, and we're out of time. So have a wonderful conclusion to your Monday. If you have lab this afternoon, have a wonderful, what is it, about an hour? A little bit over an hour, about an hour and 15 minutes, and then I'll see you once again.